Every single physical trait in every living organism is the direct output of a precise mechanistic data storage system. Nucleic acids act as the physical hardware of this system. They come in two main types: DNA, the long-term storage drive, and RNA, the network cables that transmit instructions. This flow of data, storing, transmitting, and expressing genetic information, is known as the central dogma of biology. The central dogma is entirely dictated by exact molecular geometries. To understand how complex biological life operates, we have to look closely at the chemical hardware that runs it. The foundational unit for both DNA and RNA systems is a complex biomolecule called a nucleotide. Every single nucleotide is constructed from the exact same three-part anatomy: a phosphate group, a pentose sugar, and a nitrogenous base. The variation in this system comes entirely from the geometry of that nitrogenous base. These bases are divided into two distinct structural families. The first family, Purines, consist of a double ring structure, Adenine, or A, and Guanine, or G. The second family, Pyrimidines, feature a single ring structure. These include Cytosine, or C, along with Thymine, found only in DNA, and Uracil, found exclusively in RNA. By organizing these mathematically predictable geometric shapes in specific sequences, the system creates a finite structural alphabet capable of encoding an infinite set of biological instructions. DNA serves as the primary storage drive for this information. It utilizes a specific pentose sugar in its nucleotides called Deoxyribose. The exact physical architecture of this molecule was deduced in the 1950s by researchers including James Watson and his research partner, British biophysicist Francis Crick. By constructing physical spatial models with metal plates and connecting rods, they mapped exactly how these structural elements fit together. They proved that DNA forms a double helix with two strands running in opposite anti-parallel directions. It features a protective sugar and phosphate backbone facing outward, while the fragile nitrogenous bases point inward. These inward-facing bases follow rigid rules of complementary base pairing. Adenine always pairs with thymine using 2 hydrogen bonds, and guanine always pairs with cytosine using 3 hydrogen bonds. Because the bases face inward and are locked tightly together by these hydrogen bonds, the resulting helical structure is highly stable. It is physically optimized to secure genetic data over a long lifespan. While DNA stores the code, RNA operates as the software that physically executes those instructions. RNA deviates from DNA's architecture in 3 specific ways. It utilizes a ribose sugar, it is strictly single-stranded, and it replaces thymine entirely with uracil. This single strand folds into 3 specialized functional variations: messenger RNA, transfer RNA, and ribosomal RNA. Together, these 3 coordinate to synthesize proteins. By shedding the second strand, RNA trades the long-term chemical stability of a double helix for the physical flexibility it needs to smoothly move throughout the cell and interact directly with complex molecular machinery. Before a cell can divide, it must perform a system backup. This process, where DNA makes an exact copy of itself, occurs strictly during the S phase. The S phase is the specific period in the cell cycle dedicated to DNA synthesis. Replication is semi-conservative. This means that when a DNA molecule copies itself, each newly formed double helix retains exactly 1 original parent strand alongside 1 freshly built strand. The workflow begins with helicase, an enzyme. An enzyme is a specialized protein that accelerates chemical reactions. Helicase acts as a physical wedge, unwinding and separating the double helix into 2 distinct, open tracks. Next, an enzyme called DNA polymerase moves down these open tracks, locking new complementary bases into place. Finally, the ligase enzyme acts as a structural glue, joining the sugar-phosphate backbone together. Because the geometric rules of base pairing specifically dictate that adenine only fits with thymine and guanine only fits with cytosine, the physical structure of the molecule practically guarantees near-perfect data duplication. The ultimate physical output of all this securely stored genetic data is the complex manufacturing process known as protein synthesis. This manufacturing pipeline happens in 2 steps. The first is transcription. Occurring inside the nucleus, transcription is the process where a specific section of DNA code is copied onto a messenger RNA strand. The second step is translation. The mRNA relocates out of the nucleus and into the cytoplasm. Here, it enters a ribosome, which is a molecular machine built of ribosomal RNA, where the code will be read. The genetic sequence is read in groups of 3 bases, a grouping known as a codon. As the ribosome reads each codon, transfer RNA brings over a specific amino acid that physically matches that exact 3-letter sequence. The process is governed by strict programmatic control codes. The sequence AUG will universally initiate the build with the amino acid methionine. Conversely, sequences like UAA, UAG, or UGA act as definitive stop commands, terminating the amino acid chain. Every physical protein operating in an organism is simply the downstream 3-dimensional manifestation of a 3-letter sequence stored in the DNA drive. Because the system relies entirely on physical shape, it is vulnerable to structural damage. A mutation is simply a physical alteration in the established sequence of DNA bases. A point mutation swaps out a single base, keeping the error localized, but a frame shift mutation inserts or deletes a base, which offsets the reading frame of the codons, scrambling the entire downstream sequence. These microscopic geometric errors cascade upward into severe macro-level outcomes. They're the root cause of cancers, and they act as the biological vector for RNA viruses like HIV. In clinical settings, we see the results in genetic disorders like sickle cell anemia, where a single altered letter physically deforms human red blood cells. Because exact molecular structure dictates biological function, changing or removing a single atom in a base pair can irrevocably alter an entire organism's health. Once the mechanical rules of base pairing and polymerase enzymes were fully mapped, the system could be intentionally engineered. This visual shows a gel electrophoresis scan used for DNA fingerprinting. This technique successfully reads and matches exact base sequences to identify individual genetic profiles. This understanding also led to the development of PCR, or polymerase chain reaction. PCR is a laboratory technique used to rapidly amplify millions of copies of a specific segment of DNA. By artificially controlling the replication mechanics, we can mass-produce specific DNA sequences on demand. Manipulating this biological information architecture happens at the microscopic level in laboratories and clinics around the world every single day. Biological life is an elegant, strictly governed, and highly programmable information architecture system. We just had to learn how to read the code.